Hi, I'm Arjun Srivatsa from the Computational Biology Department at Carnegie Mellon. I'll be presenting my work, A Clonal Evolution Simulator for Somatic Evolution Study Design. To briefly introduce the problem, cancer is a disease of somatic evolution. What this means is, over a course of a human's lifespan, normal cells acquire mutations which can eventually give them selective advantages and later expand in the form of a tumor. Tumors consist of cell populations with various mutations, many of which can lead to the contribution of metastasis and relapse, and heterogeneity of this kind is very important for both treating and assessing cancers. In order for a scientist to assess any form of cancer, they must design a study to measure the somatic evolution processes taking place in a cancer. To do this involves taking biopsies and later extracting relevant biological signals from the sample, usually with sequencing protocols. From sequencing, we can then perform algorithmic and bioinformatics analysis of the mutations and evolutionary processes present within each somatic evolution population. The key insight to every form of these studies is that no singular experimental protocol gives a perfect insight into the somatic evolutionary system. This often means that many modalities are combined with various trade-offs between those modalities. To briefly overview the vast base of study design considerations, we have experimental decisions, which include the types and numbers of biopsies, biotechnology decisions such as depth, error rate, or things like whole genome versus whole exome sequencing. We also have underlying biological variability in the somatic evolutionary process itself, and then a number of downstream informatics options with which to assess the sequencing readouts. So the main question then that we are trying to answer in this study is how we allocate resources to design any particular study for any hypothesis about somatic evolution. And this is a question that has not been adequately answered by prior simulators, which mainly focus on producing readouts of particular processes like spatial distributions or other mutations present in a somatic evolutionary process. Our approach to answering this question was to develop a form of simulation software which maps from various experimental decisions, somatic evolutionary processes, and sequencing modalities into raw DNA reads. We modeled each form of the somatic evolutionary process using various stochastic assumptions. We modeled the experimental decisions using repeated independence and algorithmic properties, and similarly, we used the same probability assumptions for the sequencing technologies. Each of these were combined in various fashions to create a final DNA sequencing readout, which could then be analyzed by various informatics software. To prove the efficacy of our simulation software, we tested it for a study design problem in recovering structural variants and single nucleotide polymorphisms. That is, we simulated 75 independent study designs over a grid search space, including read length, error rate, whole genome versus whole exome sequencing, and varying coverage. And we did this for 25 independently sampled tumors with the assumption that every biotechnology option had fixed cost. What we can see here is that we found an optimal study design for this problem, having 2,000 read length, 0 error rate, with whole genome sequenced, and 25 depth coverage. Generally, we saw that there was a positive correlation between read length and score, as one would expect. We saw that whole genome sequencing recovered more variants than whole exome sequencing, and we saw differing effects with error rate and coverage, though there were some general trends with better coverage having better score and lower error rate having better score. There were also independent insights, such as ensembling, which is generally thought to improve scores, also held true in our case. Another issue that we saw was how structural variants could impact the accuracy of any variant calling report, and there was significant error rates because of highly variable structural variants in our samples. In the future, we plan to use the simulator to explore the study design problem in a more general case for flexible hypotheses and study design choices. In particular, we aim to test multi-omic technologies such as combinations of various study designs and their combinations with one another. We also aim to use ideas from Bayesian hyperparameter optimization to eventually solve this problem in the more general case And this is a particularly challenging problem in high-dimensional optimization because we are only able to query points from our simulator, and we have to use these points to make an assumption about the underlying functional structure, which is represented in the following optimization problem and pictorially represented in the bottom figure. Finally, I'd like to thank my advisor, Russell Schwartz, and Haoyun Lei, who contributed to this work, as well as all the other members of the Schwartz Lab. Thank you for listening.